Welcome to Week 5, Chapter 12. In this presentation, we're going to discuss another cell division process. This time, instead of mitosis, which we covered in Chapter 11, we'll be covering meiosis, which is Chapter 12. The major difference, which I brought up in the last presentation, is that in meiosis, you're forming daughter cells that are no longer identical to each other in genetic composition. They are also no longer identical from the parent cell from which they were taken because their genetic material has been reduced by half. And the reason their genetic material has been reduced by half is because through the process of meiosis, we form gametes, and to make a new individual, whether it's a plant or animals, the gametes must come together to form a whole individual. So again, in this chapter, we will be discussing the process of meiosis. So let's get started. Since we're talking about the process of meiosis, which is in essence gamete formation, it's important to have an understanding of where these gametes are formed. Now, if you look at the image to the right, which is of a flower, flowers are the reproductive structure of some forms of plants. And there are all different types of flowers, but in general and simplicity terms, the flower contains both male and female reproductive parts. Now, not all flowers do, but in this illustration, for simplicity's sake, this flower does. So the male parts of the flower, which this part would be the filament, and that would be the anther. In the anther, there are certain specialized cells that are going to undergo meiosis to produce sperm. And then in the central part of the flower, all the way down into what's known as the ovary, you will have the egg, the female gamete, being formed through meiosis. The cells outside of the anther and the ovary are diploid cells, and they're not going to undergo meiosis. Again, we only want the cells that are the gametes to undergo meiosis. And then over to the left, we have the reproductive systems of both the male and the female. And in both male and female, there are certain cells that will undergo meiosis. In the female, those cells are located in the ovary, and they're known as oogonia. In the male, those cells are located in the testes, and those are known as spermatogonia. In other areas throughout the males and females, there are diploid cells, but those diploid cells are not going to undergo meiosis to form gametes. So again, there's two forms of cell division. Mitosis is taking one cell and forming two daughter cells, and those two daughter cells are identical to each other as well as the parent cell. Whereas in meiosis, from a parent cell, you end up with daughter cells that are not identical to each other. They're not identical to the parent, and they have half the genetic material that the parent cell had. So let's look in a little bit more detail at the genetic material so that we can then get into the phases of meiosis like we did with the phases of mitosis. So if you recall, I mentioned that in humans, there are 46 chromosomes arranged in pairs. So in all of our cells, we have 23 chromosomes in pairs. So for example, me, if you look at one of my cells, I have chromosome number 1, and I have a chromosome number 1 from my mother and a chromosome number 1 from my father. And then chromosome number 2, I have one for my mother, one for my father, all the way down to chromosome number 23, which are my sex chromosomes. And since I'm a female, I received an X chromosome from my mother and an X chromosome from my father. So my 23rd chromosomes are XX, or a female. If you're a male, you would have received an X from your mother and a Y from your father. So you would be XY at the 23rd chromosome. So in this illustration and throughout this presentation, The genetic material that is blue is from the father, and the genetic material that's the pink is from the mother. Now recall, before cell division can occur, or in essence, it's probably better to say nuclear division because we're dividing the genetic material. Before that can occur, we have to double our genetic material. We did that with mitosis, we do that with meiosis. So in this illustration, we have a blue chromosome, one double-stranded DNA, and another blue chromosome, another double-stranded DNA. They are held together at the centromere, and each blue chromosome, each section of the double-stranded DNA. So let me clarify. This whole thing is a chromosome. This is a chromatid, and this is a sister chromatid. So the sister chromatids are held together in the middle by a centromere, and they are identical to each other, each one being made up of one double-stranded DNA. Together they make what is called a chromosome. And we can see here homologous pairs have aligned. So we can see underneath the blue the red. So let's just pretend this is chromosome number 1. So this would be chromosome number 1 in a replicated state from my father, and then chromosome number 1 in a replicated state from my mother. Here we can see the homologous chromosomes have separated. So again, this whole thing is one chromosome made up of two sister chromatids held together at a centromere. This whole thing is one chromosome made up of two sister chromatids held together at a centromere. And they are homologous to each other. Homo again means same, logos means word. If we were to continue in the division process, we would separate those sister chromatids from each other. So again, here this would be made up of one double-stranded DNA. This would be made up of another double-stranded DNA, and they should be identical to each other because they were formed through synthesis of interphase. They were held together back here by a centromere, and they were called sister chromatids when they were in that state being held together. Now that they are separated, we can look at each one as an individual chromosome. And the same applies down here for the pink ones. So just kind of a quick overview. Interphase occurs in the cell cycle for both cells that are going to undergo mitosis, those would be your somatic cells, and cells that are going to undergo meiosis, those would be your germline cells, or the cells that are destined to become gametes. The cells that are destined to become gametes, because we want our newly formed cells to have genetic variability and they need to have half the genetic material, they are going to need to divide twice. So we have a first part of meiosis called meiosis 1 and a second part of meiosis called meiosis 2. Notice within both meiosis 1 and meiosis 2, we have our prophases, our metaphases, our anaphases, and our telophases.